Hello, everyone, and welcome. I'm Marie Johnston, Education Program Manager here at the American Society of Agronomy, and it is my great pleasure to introduce today's webinar entitled Challenging the Status Quo, Starter Fertilizers and Biostimulants. We've got two speakers joining us here today, as you can see, but before I introduce them, I have a few things to announce. As usual, I've got a list here to go through. It'll take me 2 seconds. First, if you need any technical assistance during this webinar, you can reach out to our support center. They are here and standing by to assist you if you can't see your screen, so you can hear my voice. Their phone number is 608-273-8080. Second, this webinar is worth one continuing education unit in nutrient management or professional meetings, depending on your certification. Your CEUs will be added to your account once you've completed the webinar or watched the full recording, so you don't have to do anything special, just enjoy the content and bring your questions. Third, send us your questions at any time during the presentation. Use this time to interact with our guests and ask them your burning ideas and see what kind of content they can share with you and their insight. Fourth, note that the webinar is being recorded. You will receive an email when the recording is available. There's a lot of information that's going to be presented today, so note that you can go back and take your time and look through it in more detail. And finally, look ahead to future events and reach out to us if you have some topic suggestions. There's also going to be a short survey after the webinar where you can give us some feedback. So take that opportunity to help direct what we do next. I also would like to, at this time, thank ICL Growing Solutions for their sponsorship of this webinar. Remember that event sponsorship lets us provide this educational content to you free of charge so that you get quality, science-based information that's unbiased and timely, and you also get CEUs if you have a certification. So please take note of our sponsor today and know that they are committed to updating you with the latest information out there. In this case, some good data about starter fertilizers and biostimulant products. All right, we have our two guests, as you can see, Drs. Eduardo Cancellier and Jason Haegele. Eduardo is a global biostimulant specialist and agronomist at ICL, where he develops foliar fertilizers and biostimulants for South America. Is that primarily where you're working now? Yeah, okay. Eduardo works to strategically position products in connection with development and pedoclimatic environments. He holds a master's and doctorate degree in soil science from Lavras, Brazil, and was honored with the IPNI Scholar Award in 2016. Joining Eduardo, please welcome Jason Haegele, who is the North American lead of agronomy at ICL. Jason leads their research collaborations with universities and third-party research contractors to demonstrate how products work on a broad range of row crops and specialty crops. He holds a master's in crop production and physiology from Iowa State and a doctorate in crop science from University of Illinois, and he is also a CCA. So welcome to you both. Thank you for sharing your work and insight with us today. Let me take your pictures off the screen and get you started as presenter. So Jason, that should prompt up to you here shortly.
Yes. So.
All right.
All right. Thank you, Marie.
You're all set. Screen.
Thank you to everyone for joining us today as we talk about starter fertilizers and biostimulants, which is a topic of great interest for me. And thinking about biostimulants, a topic of intense focus in the industry, currently all across the globe. And I'm really honored to have a great colleague with me today, Eduardo, who leads our biostimulant agronomy program for ICL, based in Brazil, but working globally now. To understand how to best position biostimulant products for different crops and different environments, all across the globe. So just a quick word about ICL. ICL is a fairly unfamiliar company in the North American and US market, but we're definitely not a new company. We've been around now for over 100 years, having started 100 years ago in Israel at the Dead Sea or near the Dead Sea. With the intent of extracting potash and other valuable minerals like magnesium and bromine from the Dead Sea, and that certainly continues to be an asset for ICL. But over that 100-year history of development for ICL, the company has expanded into a variety of specialty minerals areas, including agriculture and fertilizer, which we'll focus on today, but also industrial products like fire retardants and lithium iron phosphate batteries, and also a major presence in the food market as well. So today we're going to talk about starter fertilizers, which is a timely topic as we think about spring, which is occurring now in some parts of the country, but rapidly approaching in the rest of the U.S. and Canada as well. And I think many are familiar with starter fertilizers, or the concept of starter fertilizers. But let's start with the basic definition of a starter fertilizer. And essentially, when we talk about starter fertilizer, we're talking about a management approach, a management decision to improve seedling emergence for a wide range of annual crops, in particular row crops like corn and soybeans, but also crops like potatoes as well, to improve early season vigor, and perhaps more importantly, promote strong early season vegetative and root growth, which sets the trajectory for high yield and high productivity throughout the remainder of the season. Starter fertilizers can be applied in a variety of different methods, and so today we'll talk about some examples of trial results that used in-furrow application on potatoes specifically, but certainly starter fertilizers have expanded to other application types like 2x2 banding and similar types of approaches to placing nutrients, as well as other early season growth promoting or nutrient use efficiency strategies like biostimulants close or near the seed. Liquid fertilizers are most commonly used for starter fertilizers, although there are some dry starter fertilizers in some parts of the United States, and in the past that was potentially more commonly used. When we think about examples of liquid fertilizers, these include things like 1034O from a commodity fertilizer perspective, and then orthophosphate sources like 918-9 and similar liquid blends to supply that early season nutrition. So as we think about some of those liquid fertilizers, 1034O and 918-9 as an example, the phosphorus concentration or phosphorus analysis of those products is highest. And so the question is, why has the focus been on supplying phosphorus in starter fertilizers, even though they may contain other nutrients like nitrogen, potassium, and micronutrients as well? And I think the concept here is that, through soil testing and other approaches to managing phosphorus fertility, soil phosphorus is oftentimes adequate, based on the soil test values, but we can still experience early season deficiencies, based on, the reduced or limited phosphorus diffusion that occurs in cool, soil temperatures, as well as the effect of, cool springtime temperatures, both air temperatures and soil temperatures on, plant growth in general, and how that limits root growth. Because phosphorus is primarily taken up through diffusion, we know that root growth and root volume in the soil is important for maximizing the amount of root area that is close to nutrients to minimize that distance over which the nutrient diffuses. And so when we have low root length density, particularly early in the season, the proportion of the soil that is being explored by the plant is relatively low. And so through using starter fertilizers, whether that's in furrow, close to the seed, or placed off to the side of the row, we supply a higher concentration of nutrients close to that developing root system to try to overcome some of these early season phosphorus deficiencies or limitations. Another factor that plays into cool springtime temperatures in the U.S. and Canada in particular is that soil temperature definitely has an influence on the emergence rate of annual crops like corn. And so this example on the right is showing the mean days to 50% emergence for corn as a function of the average daily soil temperature during emergence. And so essentially temperatures about 57 degrees and cooler have a significant effect on delaying emergence, whereas when we get to 60 degrees or above, the plants emerge from the soil relatively quickly. And so this becomes a challenge as we push planting dates earlier and earlier, in that the plant is going to typically take a longer period of time to emerge from the soil due to those cool springtime conditions. Nutrients, starter fertilizers don't necessarily impact that that rate of emergence, but there are other components that can be added to starter fertilizer blends, like plant growth regulators. Gibberellic acid is a good example that can improve speed and uniformity of emergence, and potentially biostimulants have a role to play in this as well to improve the speed and the uniformity of emergence in those challenging cool springtime conditions. The other thing to take into consideration is that soil temperature is certainly a function of the weather, the climate, and air temperatures. But the soil temperature that a seed or seedling experiences can vary spatially across the field for various reasons, including variability in seeding depth, soil characteristics, soil color, soil moisture, etc. That lack of uniformity that can occur in cool springtime conditions, and the lack of uniformity in emergence, and then plant development that occurs after that can sometimes reduce yield potential. And so here's another conceptual example showing the effect of essentially planting delays, but also mixed stands of seedlings emerging at different times, and the impact that can have on yield potential of a crop like corn in this example. And so these reductions in yield potential are often thought of as coming about through competitive effects from plants that emerge more quickly versus those that are slower in their emergence. But also this lack of uniformity in emergence can lead to variability in response to agronomic management decisions that come later in the season. So for example, if we think about a fungicide application and potentially the importance of optimal timing of fungicide application for crops like corn, having different portions of a field that emerged at different times can lead to different responses, or in some cases negative responses to fungicides or other products that are being applied later in the season. All right. So thinking about starter fertilizers and this question of what is the status quo, I spent several years working in the field as a commercial sales support agronomist, and late winter, spring, when starter fertilizers were about to be applied, there was always a lot of questions about concepts of orthophosphate versus polyphosphate, the cost, are higher quality, high ortho products, are they worth the additional cost? What is the effect of salt index of different starter fertilizers? And so I think one aspect of the current status quo is these questions of cost. Is a starter fertilizer worth it? Is an orthophosphate truly superior to a polyphosphate starter fertilizer like 1034-0? What is the effect of salt index or high salt index products versus low salt index products on salt sensitive crops like soybeans? These are all important considerations. I would also say that based on my experience in the field, that a common perception is that starter fertilizers don't work consistently, or that they're only needed in specific environments like early planting or cold northern climates. And so I think in a way that's a founded concern based on the previous slides that I've showed, highlighting the importance of rapid uniform emergence, particularly in cold challenging environments. But as we'll highlight today, there are many other products now besides just nutrients that can be applied early in the season as a quote unquote starter application, things like biologicals, biostimulants, and micronutrients. And so we need to think about the challenges and the unmet needs that occur in these different fields, different crops, and different environments, and really tailor the products and the mixes of products to address those agronomic challenges that are going to be most likely to occur in a particular environment or field. The last point that I'll make here is that as we consider liquid starter fertilizers, and particularly those for phosphorus, common use rates are usually 3 to 5 gallons per acre across a range of crops. In some cases, it can be higher. Potatoes, as an example, can often use higher rates of liquid starter fertilizers. But the question, and to be honest, I don't have a great answer to this, or I haven't come to a conclusion. But my question that I would pose to the audience is: are these rates of 3 to 5 gallons per acre optimal for just the common fertilizer sources, or is it just simply the way that it's always been done without a strong scientific or data-backed reason to support these 3 to 5 gallon per acre rates? When we choose between phosphorus starter fertilizer sources, again, the question is often polyphosphate sources like 1034-0 and 1137-0 versus orthophosphate sources. And there are different considerations that are involved. Seed safety, polyphosphate, 1034O in particular, has a higher salt index. Seed safety is going to depend on the rate used, soil characteristics, crops to an extent, corn versus soybeans, etc. There are certainly considerations of compatibility. Polyphosphates are going to be more compatible with cation nutrients like calcium, magnesium, and micronutrients, whereas with orthophosphates, chelation, EDTA chelation for micronutrients as an example is probably going to be more more required or needed by that fertilizer source. And then lastly, there's the consideration of cost. Lower cost for commodity products like 1034O versus higher cost potentially for specialty or semi-specialty liquid starter fertilizers like 9-18-9 and similar products. So here's an example of a starter fertilizer trial, and this is on potatoes. Switching gears from corn and use potatoes as an example now. And in this trial, we had 2 geographical locations, one in Wisconsin and a single location in Michigan, but with 2 different potato varieties. In each of these locations, we had a control treatment. So control treatment had no in-furrow application of nutrients. It would just be the grower standard practice of dry broadcast N, P, and K. We had ammonium polyphosphate or 1034O applied at 10 gallons per acre as a grower standard liquid starter practice. And then an ICL product, Agrolution pHLow, applied at 10 pounds of dry product per acre. So this is a water-soluble fertilizer solubilized into approximately 3 gallons of water per acre and then applied in furrow. If we look at the Wisconsin location, there really wasn't much of a starter response there. Treatments weren't significantly different. And then at the Michigan location for both varieties, we had statistically significant increase in potato yield, marketable yield expressed as 100 weight per acre for applying starter fertilizer, either source. And then we would conclude that based on the statistics, the 2 different starter fertilizers were not different, even though there was a numerical advantage to one versus the other. But the question I would pose is, does this data actually tell the whole story? So as we go on to the next slide and compare these 2 different products, again, this Agrolution pHLow nutrient source is a water-soluble fertilizer, has a guaranteed analysis of 11-45-11. So at that rate of 10 pounds of dry product per acre, we're applying only 4.5 pounds of P2O5 per acre versus nearly 40 pounds of P2O5 per acre in the 10 gallon per acre rate of 1034O. So almost 10 times more phosphorus being applied as 1034O versus the Agrolution pHLow water-soluble fertilizer. So to conclude this section, as we look at phosphorus use efficiency, and I'm expressing this as 100 weights of potatoes per pound of P2O5 applied per acre as a starter fertilizer, we see that there are some significant increases in efficiency from one source versus the other, even though it's applied far less phosphorus per acre. We were getting similar or potentially greater yield responses, but with approximately 1/10 the amount of phosphorus applied per acre. So this comes back to this question of, where do these liquid starter fertilizer rates of 3 to 5 gallons per acre for corn and soybeans, or maybe 10 or more gallons per acre for potatoes, why are they common? And can we challenge that status quo and consider other options, whether it's different nutrient sources like we've highlighted here, or potentially other strategies like biostimulants, micronutrients, other additives to a starter starter blend or starter product to really drive home nutrient use efficiency. So with that, I'd like to address or introduce Eduardo to take over and really take a deeper dive into biostimulants and the role that they play in crop productivity.
Right. Thanks very much, Jason, for the incredible information and discussion. Good morning, everyone. So it's a pleasure to be talking to all of you here. And to start the discussion on biostimulants, first, I prefer to start with the basics on plant nutrition, because biostimulants, they come as another generation of technologies to improve the way we do agriculture, but we should always rely on the basics of what we already do well. And I'm bringing up the concepts that you might know very well. The 4 are nutrient stewardship, where the right source, right rate, time, and place for the fertilizers play an utmost role on driving an improved nutrient use efficiency. And what Jason has just discussed and demonstrated is that choosing the right product formulation, and that right product formulation has to do a lot of the timing of application and the place, because it has all to do not with the overall soil fertility as discussed, but the placement at a specific time of demand from the plant. And we always look at those 4 R's, basically trying to improve how plants take their nutrients, to improve nutrition. And the reason that we need to look for those is that soil reactions prevent us from taking up all the nutrients available. Plant statuses might hinder the capacity of plants to take up those nutrients. It's not that the soil does not contain, but the plant has a physiological status that is preventing it from using fully. And of course, it interacts with weather conditions, leading to a range of different well-known problems associated with the fertilizer efficacy. So to the next, please. So taking into account this interaction of soils, here I want to bring it back. What is the function of soil to the plant and how it interacts with nutrition? Soil has several functions, like supporting plant growth. It's a physical role. We might, everyone know that we can grow plants without any soil in it, in soilless media, hydroponics, etc. Soil has a role on retaining water, retaining nutrients, and serve as habitat for microbiota, which is an important part of making nutrition more efficient. So if I'm to make it really short, soil serves plants in 3 purposes: to hold water, to hold nutrients, and physically hold roots. And as an agronomist, I studied my whole career, and it's the most common saying that you find in the field is that soil supply plants nutrients. That's for next. And I really want to question if that is really so, if plants supply nutrients. Because if you take a deeper look onto the relationship, soil does not supply anything. Soil, although it's alive because of soil microbiome, the interactions with plant nutrients are based on the availability of soluble nutrients on the soil solution, directly interacting with the nutrients held onto the cation exchange capacity. So soils retain nutrients, don't supply. Plants need to act onto their own capacity, on their own foraging capacity to go after those nutrients and uptake them. So we largely underestimate or not drive as much attention in the management on the capacity of the plants to take up those nutrients. Next, please. And when we are talking about the uptake capacity, the first thing we need to look at is that the soil rhizosphere, the plant rhizosphere, soil rhizosphere, which is, rhizosphere, just as a reminder, is the volume of immediate surrounding of the roots, the soil immediately surrounding the roots, in which the plant modulates the chemical and biological and physical properties. So it's the area of influence to plants. So you can look at this figure in the left here, that different elements, they interact with rhizosphere in different ways. Elements such as gases like oxygen, CO2, they move quite far away from the roots, more than 4 millimeters, while the nutrients, while the mineral elements, and I will highlight here the NPKs, usually the area of influence of the roots is roughly 3 millimeters, from 2 to 4. And that drives us to an understanding that for plant to uptake nutrients, roots must be close to the elements in the soil. And why that is relevant? Because then root growth and the volume explored, the volume of soil explored by roots, play a big role onto the capacity of plants to uptake. And here I have a second take, because sometimes we grow crops under no-till management, and one of the big problems of no-till is the potential to build up soil compaction, or even in soils under tillage, you can have hardened layers where roots may have a hard time to penetrate and access those nutrients onto that volume of soil. So whenever you have some big soil clots where roots have a hard time accessing and driving the roots through the whole volume of soil, it's a first factor of reduced nutrient use efficiency. So for management, we need first to make sure roots explore the soil through every cubic centimeter or inch, as if you prefer in America. Every cubic inch of soil needs to have a bunch of roots crossing. If you dig up your soil and your crop and you have some volume of unexplored soil, it means that you're losing nutrients and you're losing yield potential, you're losing money. Next, please, Jason. But to improve that capacity, we have ways to manage that. And I'm bringing here one of the most common biostimulant substances that helps plants to improve their capacity to uptake those nutrients. First, humic substances, exogenous applied in a concentrated manner around the root system, they have been proven to increase the biomass production. Not only that, but also to modulate the root architecture. Plants direct more energy, more carbohydrates for root growth, improving their density of root hairs, which is especially important, as you can see here on the right side figure. This high density of root hairs is particularly important for the uptake of nutrients of low mobility, highlighting phosphates, zinc, copper, and some others. Not only the root growth, but root architecture changes the way it does branching. And for last, the cell rhizodeposition, which is the way in which the plants communicate with soil microbiome. So plants can modulate through that root exudation how the microbes grow. So next, Jason, please. So those humic substances, they have been proven, and they are one of the most largely studied biostimulant substances. And by here, I want to comment on one of the very commonly said misconceptions on biostimulants. If you go out to read about biostimulants, you will very commonly read that one of the issues or problems of biostimulants is that we don't understand their mode of action and how they work. And I'll prefer to say that it's a partial truth, because some biostimulants, like the humic substances, we do understand very well what are their chemical and physiological responses to the plants. So for those more basic biostimulant substances, yes, we do know how they work and their mode of action. While it's also true that from some newly discovered substances, we might indeed not understand fully how they work. Just separate exactly what you're talking about. So those humic substances, as you can see in this figure, they trigger a range of physiological responses to the plants. So first, it changes the plasma membrane potential. But what does that mean? So it means that it activates one enzyme at the root surface level, hydrogen ATPases, and that causes two effects. First, on the left side, root growth promotion. The increased activity of those hydrogen ATPases increases the acidification of the rhizosphere. And this is part of the acid growth theory, which makes the plasma membrane, the cells, more easier, let's put it like that to make it more simple, for cell division. Hence, it will promote root growth. This is a similar mechanism in which the auxin, a plant hormone, induces root growth. And this is the reason why the humic substances are commonly said to have an auxin-like effect, meaning that it will affect the root in a similar way to that of auxin, promoting root growth. But that activation of the soil surface enzyme causes also a secondary effect. It activates ion transport enzymes, meaning that the transporters on the root that brings nutrients inside the plant will also be more active, increasing the potential not only for the roots to grow more, but to more efficiently uptake what they have in their hands. And on the bottom red circle, you see organic acid exudates, again, as a means of communicating to the soil microbes and improve the synergistic effect of the microbiome with the plant. Next, please. So coming down now to the more biological part. When we grow, especially in open field or broad acre crops, we have brought a system in which under natural vegetation is highly heterogeneous in terms of vegetation and species living there, which will eventually feed a very diverse and balanced soil microbiome. But when we bring it to monoculture or even a succession of few crops, we largely reduce the diversity of materials added to the soil, the organic materials. That in turn will also select some microbial populations which might have negative effects on the soil balance, hence being more prompt to the appearance of soil-borne diseases, and also reduces the activity of some of the naturally soil nutrient solubilizing microbes. Next, please. So biostimulants is also a mean to improve that connection of plants with soil microbes. I have here in the center a picture of corn roots. The adventitious roots, it's typically happens this type of sugar exudation on those roots around tasseling. It's when you can clearly see this large exudation, but it's not because you can see only at this stage that it happens only at that time. Crops constantly exudate large part of the carbon that they capture through the roots, and that is to feed this whole range of microbes around rhizosphere, that it will eventually help plants to solubilize nutrients. On the biologicals market, you are constantly hearing about nitrogen-fixing microbes and soil solubilized, phosphate solubilizing microbes. I need to remember you that our soil already contains some native microorganisms that do have those benefits. Okay, they are not as efficient, not as closely efficient as the one we have been selecting and applying through some microbial products, but we can enhance that activity once this connection with the plant is promoted. It's a natural process that it will happen, either you like it or not, but we can improve it through the use of some biostimulants, which will improve that connection. Next, please. So, and as an evidence of that connection, we have conducted some trials, and since this product based on humic substances have been under use in Brazil for quite a long time, we have followed up around 40 different strip trial plots, and we have done biological measurements on the root rhizosphere. We have measured the activity of 3 different enzymes that are directly correlated with the capacity of solubilizing phosphates and sulfur, but the most important part is not their direct function of those enzymes, but those enzymes will serve as an indicator of the overall biological activity. So we have seen across the 3 different enzymes increases on the order from 11 to 16% increase on the activity, demonstrating that even though the humic substances might be seen as integral part of the soil organic matter, the exogenous application can indeed promote further increased responses from the plant and the soil microbiome, which is great. Even in small amounts of those enzymes we can give you a great nutritional effect. So next, please. And as a second evidence, I want to bring you here a concept on how the biostimulants can possibly help plants on the start of fertilizer and the early development stages. So first, we discussed how the early root development and the contact of roots exploring the soil can get your elements closer to the roots, making it easier for the rhizosphere effect to take place, and that proximity boosts the uptake of nutrients. Second, plants can modulate through the root system the activity of the microorganisms on the rhizosphere level, which is also a mode in which microbials help plants to uptake more nutrients. And third, I'm gonna show some consolidated evidence here demonstrating that some phosphate solubilizing microorganisms like the Pseudomonas pseudoacaligenesis, so everyone can read in their own accent, sorry for that. It's Latin, I think no one here is speaking Latin at the moment. So in the x-axis, you can see the capacity in which these bacteria can solubilize the phosphates. So you can see measurement of dissolved phosphates in solution. So 5 different types of biostimulants have been added to the soil, and none of them barely changed the level of phosphates in the soil. The addition of these phosphate solubilizing bacteria has granted a roughly 10 milligrams per liter of dissolved phosphates. But the combination of that phosphate solubilizing bacteria together with some, let's call them activators, and among them there are some amino acids which has prompted that the microorganisms to activate their phosphate solubilizing capabilities. Let's say it's a wake-up signal. So once that microbial has received the stimuli from the threonine, not only more phosphate has been solubilized to the soil, but it has been demonstrated that the percentage of phosphate on leaves has been increased, as well as the one of nitrogen on corn trials. Of course, you can later on read the reference on the screen to understand further on, but it's a demonstration that even small additions of some organic molecules, they can trigger responses to the root system and to the soil microbiome, making plants more efficient. Next, please. Making it more visual, I have here 2 examples, one in soybean and one in corn. On the 2 pictures on the left-hand side, you have what we call rhizotrons. So we close that glass with some dark covering, which allows roots to grow against the glass and allows us to visualize how root grows on time. And you can see here soybeans at very early stages of development. And you can see in both pictures that the soybeans are really on the cotyledonal leaves, barely open the first trifoliolate leaves. Roots are already quite deep, more than 20 centimeters deep. And on the right side, we have used some biostimulants from ICL to modulate that root growth. And you can see how that plant will be much better prepared to handle the abiotic stresses, such as some type of drought, or even have an increased capacity to explore the nutrients on that soil. On the right-hand side, the corn plants, they were grown in pots so that we could wash the roots. You can see here in the plant on the left, the control treatment has a much lower root volume growth as compared to the treatments 2 and 3. And you can see here that the biostimulants, of course, we have different types of biostimulants based on amino acids, unique substances, algae extracts, etc. There is an infinity of possibilities out there, and not always biostimulation is not something like you can use an indefinite amount. So if you use too much of it or the wrong combination, you can get negative responses here. And if you compare treatments 3 to 4, you see that the shoot growth is similar while the root growth has been decreased. So choose well your right formulation for the right stimuli. Next, please. And I want to bring up the conversation of other starter fertilizers, which, in my opinion, it's a bit of a gray area of whether this is a biostimulant or not. I bring here especially nickel and cobalt, 2 elements commonly used in early stages of soybean. And now I ask you whether seed treatment can be considered a starter fertilizer or not, because commonly you see this as a seed fertilizer, which is applied at the same time of starter fertilizer, and they greatly enhance the soybean nitrogen fixation capacity. However, whether they are considered biostimulants or not, it really depends on how you look on a regulatory framework. I watched an amazing talk from Professor Patrick Brown from the University of California, discussing this, how nickel, which has been consolidated as a plant nutrient by science, proven, in some countries, regulatory-wise, the laws consider it a contaminant, such as it's the case of European Union. The new fertilizer regulation consider nickel a heavy metal contaminant, so it's not allowed under the law, even though it's a plant micronutrient. Depending on the country, or maybe under the states of the US, if you don't have nickel covered under either a nutrient, beneficial element, or a contaminant, it might as well be considered biostimulant. We might bring this element to the market as a biostimulant, because as a concept, biostimulant is a substance that brings benefits to the plants, which is not a fertilizer nor a plant protection product. So what is it? For nickel, you could argue it's a plant nutrient, so it automatically brings it out of the biostimulant arena. But cobalt is not a plant nutrient. It's an element greatly beneficiating the bacteria. It is used as part of an enzyme that transport oxygen out of the nodules, which is fundamental to keep the active nitrogen fixation process. So if it's not a plant nutrient, what is it then? A beneficial element. But if your fertilizer regulations don't cover cobalt as an element, you can only bring it as a beneficial element on the biostimulant regulations. So there's a lot of discussion whether they are biostimulants or not. People will differ in their opinions. While it's very clear what they do, I think law here plays a role on whether they can be included to the market under one regulation or the other. Some other two elements here, especially silicon, I think it's a hot topic in the US now, since new regulations have changed, allowing more flexibility to bring products into the market. Very well studied and understood beneficial elements, bringing positive responses to many crops. But again, depending on the regulation, it will not be a plant nutrient, so it is a biostimulant. For Brazilian law, just as an example, silicon is under it's a nutrient under the law. So you can have a fertilizer based on silicon. But this is not true for the European Union. In the European Union, you don't have silicon as an integral component of regulation on fertilizers. And selenium, it's another new element commonly understood as an element for biofortification, but also has had its efficacy proven for a number of abiotic tolerance, increased in abiotic tolerance for plants. So definitely under the traditional claims of biostimulants, and it's usually not covered under any regulations. So bringing it under a gray area of whether I should include selenium on a specific biostimulant product or a fertilizer, or if it's forbidden at all because it's a metal not covered under any regulation. So it brings a little bit of, a further confusion on how we could approach those products to the market, but it's more something that I like to consider as opportunities. And from the biological point of view, I like to say the law of nature doesn't care for the law of man. Doesn't matter how we treat it under the law, the plants will respond to those elements on a positive or negative way as they were. So next. So to close my part, making it long story short, all the fertilizers technology that we use, positioning the 4 R's, we aim at bringing elements available at root level. After elements available for the roots in the appropriate chemical form, it's up to the plants to uptake and use those nutrients. And this is where biostimulants take place. Biostimulants, it's about improving the capacity of plants to uptake those nutrients. And fertilizers deliver them in an efficient way at the soil solution level. While biologicals could add even further, biological biostimulants, they could add more nutrients to the system through biological nitrogen fixation, for example. The traditional organic biostimulants will improve plants' capacity. So that's my message for today. And now we'll hand it back to Jason, who has some interesting data to demonstrate that in terms of number.
Okay. Thank you, Eduardo. Really appreciate your insights and knowledge on biostimulants and maybe a different take on biostimulants compared to what we typically see a lot of times. So to close things up, I want to revisit this idea of fertilizers and biostimulants in combination, but also take a moment to pause and address a couple of questions that have come in while Eduardo was speaking. And the first of the questions relates back to the previous data example of starter fertilizers on potatoes in Wisconsin and Michigan, and I believe the question was related to background levels of phosphorus. I do know for the Wisconsin location, it was 52 parts per million phosphorus, so relatively high, and that was also a sandy clay loam, so a little bit higher clay, higher organic matter compared to the Michigan site in particular. So that may be explanatory for the lower starter fertilizer responses in Wisconsin. The Michigan site was a very low organic matter, high sand site, about 72 or 73% sand, in the soil texture characterization. Irrigated site as well, so likely lower nutrient availability, definitely lower organic matter in Michigan compared to Wisconsin. That being said, there's a fairly well-established body of literature and data for potatoes showing that potatoes respond positively to phosphorus, often in soil tests values that are already high for phosphorus, and I think that relates back to the the different rooting architecture, root system of potatoes, relative to a fibrous rooted system, crop like corn or wheat, and the implication that has for phosphorus uptake. The other question was related to active ingredients or biostimulant ingredients in the BIOZ Diamond product that has been referenced, and we'll include in this example here. This product contains fulvic acid and a yeast extract, so fairly high amount of fulvic acid along with yeast extract, which is likely contributing amino acids and other bioactive molecules, and then a nutrient analysis of 10-01 as a soil application. So in this particular example, kind of continuing on that idea, this is the same trial that I introduced at the beginning of the presentation, comparing 10-34-0 and Agrolution pHLow, and now we're introducing this biostimulant BIOZ Diamond as a additive to the starter fertilizer. The biostimulant will be applied at 1 quart per acre, the recommended application rate, and so that you cross these different comparisons and 2 different starter fertilizer sources, different potato varieties, the addition of the biostimulant resulted in an average increase of 3,200 weight per acre. So there was certainly a yield increase, and we've seen that on other crops as well. Same type of trial comparing 10-34-0 and Agrolution pHLow for corn and soybeans, where we add the biostimulant, the BIOZ Diamond, and we have an increase in yield. But what I'd like to focus on is actually the improvement in phosphorus fertilizer efficiency. And so if we again calculate P fertilizer efficiency as 100 weight of potato increase per pound of P2O5 applied, we have a slight increase numerically when we add the biostimulant for both starter fertilizer sources. This increase in efficiency is small compared to the efficiencies that I showed earlier in the presentation for the selection of one starter fertilizer source versus another. But I think this reiterates the point that when we use biostimulants, particularly when they have yield improving effects, we can also either directly or indirectly increase nutrient use efficiency for nutrients like phosphorus in this example, but also other nutrients as well. Kind of the idea that any increase in yield that results without an increase in fertilizer application rates is in effect an increase in fertilizer use efficiency. So in conclusion, starter fertilizers, and I'm using that term loosely now, create the foundation for greater nutrient use efficiency and productivity. And starters is in quotes because we've talked about starter fertilizer sources, for phosphorus specifically, but now with different additives, biostimulants, some of the micronutrient or trace elements that Eduardo talked about, I believe it's possible to think about new ways to use starter fertilizers for a broad range of crops, not just potatoes, but corn, soybeans, and other annual crops to achieve different outcomes and improve yield and nutrient use efficiency. And as Eduardo has done a really nice job of, biostimulants interact very nicely with nutrients and essentially in many cases improve the capacity of plants to either take up the nutrients or use those nutrients through enhanced internal physiological processes within the plants that allow a crop to convert that nutrient fertility, that nutrient fertility to a harvestable biomass like a grain or a potato or a forage. Thank you for your time today on behalf of ICL Growing Solutions, not just here in North America, but globally. Eduardo based in Brazil and myself in the United States, and certainly many colleagues within ICL all around the globe working daily to work alongside retail distributors and farmers to solve their unmet agronomic challenges with unique products and solutions that we create through our internal innovation efforts within ICL. I invite you to contact us directly if you have questions after the webinar, or for those who watch it later. We certainly have a website here in the United States that has trial information, product information, and a lot of also very useful agronomic thought leadership pieces. And if you want to get in touch with us to continue the conversation about starter fertilizers and biostimulants, you can also use the QR code there in the top right of the slide. So with that, Marie, I'll kick it back to you, and you can wrap up, or if we have some additional questions that we need to address, we can certainly do that now as well.
Yeah. As you've probably been seeing, there have been a number of really good questions that have come in. I think something that they kind of relate to, so you presented a number of slides with data that had variability about the same magnitude as the difference that you saw. So maybe you could spend a little time talking about irrationalization or the explanation for why these biostimulant type products have such variability, and maybe what we could do to hone in on what works in which situations.
Mm-hmm. Yeah, I think that's a great question, and I ask Eduardo to weigh in on this as well. But I've been involved in biostimulant research and trials now for a number of years, and other classes of similar type products like plant growth regulators. And one of the things that I've learned over the years is that these products don't always work consistently in every year in every environment. I think that can be attributed to a number of different reasons or sources. But as we're gaining more experience with biostimulants, different types of biostimulants, both microbial and non-microbial, and humic acids, fulvic acids, seaweed extracts, etc. I think it's clear that these products have different roles to play, different different uses for different crops, different application types, foliar versus soil. And as we gain this knowledge over time, it enables us as an industry and as crop advisors to more intelligently, more effectively recommend the right types of products for the right applications, and also realize that depending on the year, the weather, the environment, it may be a decision that is worthwhile, or something that we need to consider carefully, and maybe have a different approach to how we manage the crop, depending on the environmental conditions that particular year presents. So I think it's a very important question for for ICL, but also for the industry as a whole. And Eduardo, I'd also like to get your take on it as a kind of the global perspective on biostimulants.
Yes, thanks, Jason. And I agree with you. And it's one of the challenges of the use of biostimulants in agriculture in general is about this inconsistent, not as consistent result as we have for the pesticides or for the fertilizers. And the big problem here lies on the lack of our capability to diagnose all the environmental factors related to the response of biostimulants. We discussed, for example, on how humic substances, they could possibly activate some physiological processes at root level, but those processes could be naturally being boosted by natural factors. And even someone mentioned on the question how biostimulant performance under high soil organic matter. So while soil organic matter is different from an organically added molecule, we can't exactly know the exact environment where this response will be greater or lesser. What we know is that on average, the response is quite positive. In most of the cases, it works, but indeed, in some not. And when we talk about biostimulants, we are talking about a huge variety of different molecules and possibilities. The definition of biostimulants is sometimes is not based on what it is, but on what it's not. So if a substance affects the plant growth and the root rhizosphere microbiome, not a fertilizer, not a crop protection, it's a biostimulant. So anything, there is like hundreds, if not thousands of different molecules or elements we could consider biostimulants. So it's really difficult to generalize anything on biostimulants, and the lack of diagnostic tools is one of the problems, and it's a problem that I don't see a solution soon, because if we are talking about the application, the external application of any amino acid, how can we possibly know that this amino acid will perform better? Can we really test the plant, a leaf test for the amino acid or the whole 20 amino acid levels every week into our plants to understand how it's performing according to the soil, according to the weather, according to the different varieties? I cannot see us being able to map that so soon. So that's where the challenge lives. So that's how we base ourselves on data on a large amount of trials like the ones presented here, that the average is usually positive, and it serves for the farmer as a type of insurance, making sure that the right physiological process is active.
Yeah. So something that I was thinking about pretty early on in the presentation, Eduardo, you talked quite a bit about, well, not quite a bit, but you mentioned how root interception, like getting the roots where the nutrients are, and like that whole microbiome influence. So I was wondering if you could talk a little bit about like water management or nutrient management, because certainly you don't want to, there's a balance, I imagine, between stressing the plant out and getting the roots to explore, but also not stressing the plant out. So do you follow my thought? Like, how do we encourage the plant roots to be where we want them to be?
Well, from here we have two very different approaches. One is irrigated agriculture and rain-fed agriculture. So while for rain-fed agriculture, you want to maximize the root depth in order to prevent abiotic stress associated to drought, to dry spells. So there is not much to talk about water management when you talk about rain-fed. It's just preventing, preparing your plant to handle better times of lack of water. While in irrigated agriculture, you don't have as much as needed to improve root growth, because roots will be confined to the wet bulb. If you are talking about irrigated agriculture in arid regions. If you're talking about irrigation as a supplementation, then again, you will still want the roots to go deeper to suffer less with water drought. So unless we go into very specific condition, it's hard to discuss. If you go to a condition of arid conditions where the roots are restrained to the wet bulb, the benefit of humic substances will be a secondary benefit, not on root growth, but more on the activity of roots and in the microbiome. So you only do have the physiological benefit, not the physical benefit of foraging capacity. So it's still positive, but not to all of the process.
Yeah, okay. That's fair. So then what are your thoughts on the future directions in this whole arena of products? I mean, do you see like branching of different kinds of products or suites of products, and maybe what's in them or what they're intended to do?
Yes. First, again, the biostimulant is a generic term that can mean a lot of different things. And products, they're in most of the cases specialized on what they do. So while what we have discussed today has to do with improving nutrient efficiency or uptake of fertilizers, other biostimulants will handle abiotic stress or will improve the development of color in fruits. So yes, there is a branching, And the tendency is that biostimulants, they are not classified based on their composition, but on their function. That it's also part of the definition, even in the law. The law states the claim the product has. And usually, regulatory-wise, you need to prove or demonstrate what the product does, not exactly their composition, as opposed to fertilizers. Fertilizer is about composition, not the effect.
Mm-hmm. Yeah, I think the thing that I would add is that, we consider, simpler biostimulants now, humic acids, fulvic acids, seaweed extracts, these types of biostimulant sources have been around for a number of years, and in some cropping systems and some parts of the U.S. or globally are more established compared to other cropping systems. But I anticipate that as the knowledge and experience with biostimulants continues to grow, we'll start to see more sophisticated combinations of these basic ingredients to solve very precise agronomic challenges or specific functions in the plant that we want to affect. So, for example, Eduardo mentioned increasing color development in fruit, fruit like grapes or maybe apples or cherries, and so putting together the right building blocks to affect that desired outcome. But also, I think we'll start to see other types of technologies start to enter the biostimulant space. So things like RNAi and other technologies that on the surface make people very very nervous, but as we learn more about the genome of both plants and the microbiome as well, plant physiology, microbial physiology, there's going to be lots of opportunities to take advantage of that knowledge to develop very precise, targeted biostimulant technologies for different crops and different outcomes.
Okay, so we're at the top of the hour. I just have one quick question. Maybe you have something you can suggest here for our audience. So where would you send people for more information about these types of products? I mean, certainly the scientific literature is one place, but is there like a forum someplace out there? Or a similar type platform that's evolving as these materials become on the market?
What
suggestions might you have, Eduardo?
Biostimulant is very complex. If you look out there on scientifically wise, you have a lot of materials explaining biostimulants, but you can always get in contact with us if you have a specific interest in some crops. I can direct some targeted reading. While in ICL, we do try to publish several more easier to read papers, I would say more directly to the farmer knowledge. You can always consult the website, ICL website, both the American website and the ICL global website, but contact us through the link and we'll give you some hints directly based on your interest.
Okay. But thank you, Eduardo. Very
good. And yes, there's a short little survey that'll pop up for all of you that are attending today. Thank you to both of you. It was a fantastic presentation. I think feel like you've opened up more questions than maybe you had anticipated. There seems to be quite a bit of unknown in this area. So thanks for everyone attending, and watch for, maybe we'll return to this topic in the future. I imagine that we probably will in the fall. So thanks again. And I hope that you found, and I hope to see you on the next one. So enjoy the rest of your afternoon.
Okay. Thank you, Marie. Thank you, everyone.
Yep. Thank you, everyone. Bye-bye. Great to have you today.
Bye.